Hi there, friends. Welcome back to Little Lab at Home, Little Lab / Lab at Home with the Museum of Life and Science. I'm happy as always to be here with you along with Catalyst volunteer Caroline, who's going to be helping us with today's experiment. I hope that, as always, you will all help lead our program with your thoughts and questions and ideas in the chat. So this week, we are continuing our Black Excellence in Science series to highlight the work and contributions of historical and current Black scientists. This week and in the coming weeks, we are going to be highlighting Black scientists who are doing their work right here and right now in the Triangle, because Black excellence is not just historical, it is of course current. I just wanted to be able to show you kind of a few pictures of who we're going to be talking about today, as well as kind of some other things about what we will, kind of some subject-related pictures. So I'm going to ask you to pull those up, Caroline, if you would.
Can do.
That looks great. All right. As this slide suggests, I am going to be introducing you all to a friend of mine and to a scientist who is very excellent. Her name is Dr. Christine Daniels. She is a cell biologist and a molecular biologist, as well as a postdoctoral associate with the Duke Human Vaccine Institute. She studies human disease, including cancer and viruses, and she studies them at kind of the tiniest level, looking at the ways that viruses and cells interact chemically. Her research is an important part of developing different kinds of medical treatments for people who are living with viral diseases. Let's go ahead and check out our next picture here.
Sounds great.
So this is, these are some pictures from programs Dr. Daniels has done with us before. Dr. Daniels is, because she's also an amazing science communicator, she's helped us understand viruses in our Parenting in a Pandemic webinar, along with answering Lab at Home viewer questions in a special Q&A session for our Safer with Science series. Try saying that 5 times fast. You can hear more from her in those programs, which we will be linking in the chat here in just a few minutes. She also did a really wonderful feature for the People Science podcast, where she both goes into a little bit more depth about her research on HIV, and she shares her insight about how racism impacts the STEM field. So I really enjoyed listening to that. I hope that once we link that, we you'll be able to check that out as well. Finally, we'll also include a link to the Duke Human Vaccine Institute website, which is a great place to learn more about her work and the work of other cell and molecular biologists like her. It's a bunch of really exciting stuff, so we'll be linking that as well. I think let's go ahead to our next slide here if we could. Beautiful. Before we get to our experiment today, I want us to take a quick look at this microscopic world that Dr. Daniel normally navigates. Much of what she studies are kind of the certain like chemical features that viruses have and how they affect people's bodies and how they make them sick in order to help make them well. What do you all know? Let me ask you all this. What do you all know about viruses? What do they make you think of? Or maybe are there any facts you know about them? This is a great contribution. We have someone in the chat saying that they infect people. That is a really good contribution, right? We know that viruses are something that can infect people. They can get passed from person to person. We have a friend saying that they make you sick. Yeah, I would say definitely. Viruses are something that can make you sick. What else? Are they very, I think we kind of said it, but are they very big or are they very small? What do you think? Oh yeah, they are very, very small. Yeah, exactly, my friends. And we'll be talking kind of about exactly how small. But I wanted to show you these pictures because they are really good. Oh, first I wanted to shout this out in the chat. We have friends saying that they can both mutate and change, and they can affect our lives, definitely, for sure. These are some different, like actual pictures of some different kinds of viruses because there are many different types of viruses in the world. These pictures were taken with a scanning electron microscope because viruses, as we said, are very, very small. They come in kind of different sizes from about 20 nanometers to more than 300 nanometers in diameter. So 20 to 300, so they have some different sizes, but they're all very, very small. A virus like one we are probably pretty familiar with, like a virus the size of the coronavirus, is kind of several thousand times smaller than a grain of salt. So if you're thinking about looking at a grain of salt and kind of if you could split it up several thousand times, that might be the size of one coronavirus particle. When we're able to see what these viruses look like through a scanning electron microscope, they look like this, which is to say, since they're smaller than the wavelength of visible light, they don't have a color. So because light doesn't really bounce off them, because they're smaller than the wavelength of that light, they just kind of look, I guess, black and white like this, right? They don't really look like they have colors, but we do have some other pictures of diagrams which show it a little bit better, kind of what we'll be looking at. So these are actual pictures, and I'm gonna, yeah, perfect, have Caroline switch to some pictures of diagrams of different types of viruses. Something that all viruses have is DNA or RNA. Do you guys know what DNA or RNA is? Does that sound familiar? Does it make you think of anything? Do you know of anything else that has DNA or RNA? What do you think? What do you think, Caroline? Do you know something else that has DNA?
Sounds oddly familiar. I think we might have DNA.
I think you're exactly right. And we have friends in the chat saying that humans do. We do, exactly. We have DNA.
Great.
DNA is what makes you, you. I love this. DNA is kind of like the recipe for how to make whatever type of living thing it is. So all living things have DNA inside of their cells. And viruses also have DNA or RNA. RNA is like if you have just half of a strand of DNA, we would call it RNA. But we would consider viruses alive. And we'll talk about why in just a second, right? All viruses have DNA or RNA, which is the kind of chemical code, the genetic code that lets them make more of themselves, and they have to use a host's cell. So we had a friend saying that they infect people, right? Viruses can also affect animals, right? Or all kinds of different kinds of animals. So a virus is something that we don't consider to be alive because it needs a host cell in order to make more of itself and in order to perform any of the functions that it does. So kind of confusing, but all living things have the DNA and RNA, and viruses also have DNA and RNA, even though they're not technically alive. You can see that there's some different kind of shapes that viruses take. All viruses have to have some sort of capsule, some sort of something to hold their DNA or RNA, their genetic material. And sometimes this looks like a spiral, like this kind of pinkish one you can see down in the lower left corner. And sometimes it looks kind of like this interesting 3D shape, this 20-sided shape called an icosahedron, and that's kind of what this kind of more greenish picture is. That's kind of the shape that these capsules take, that capsules that hold that genetic material. Sometimes it even has a tail, like our bacteriophage, which is up in the kind of upper left corner. So sometimes they have a tail, but still has that capsule, and they all still have that DNA or RNA inside of it. Some viruses, like the viruses that cause norovirus and polio, these are really simple ones. They're just a capsule of protein with the genetic material inside of them. So they would look maybe a little bit more like the kind of greenish colored ones. They're really kind of more simple. Some of the viruses that cause diseases like measles or hepatitis or the flu or COVID-19 have an extra layer of protection that is called an envelope. And we're going to be talking about that envelope today. one last picture here. And while we're calling that one up, we have a really interesting question in the chat about how viruses move. Do they, like, we have a friend saying, do they fly? And if they do, how do they do it? That is such a good question. Caroline, I love that question. Viruses do have to travel, right, from host to host. The way that a lot of them do that, they have to kind of ride on things like droplets, right? Like, for example, COVID-19 is something that has to ride on kind of water droplets produced by the human body when we sneeze or when we speak. That's a really great question. So they kind of, we could say that sometimes they're left on surfaces. Like if somebody sneezes on a doorknob, they could be left on that surface.
Then they ride your hand.
Exactly, and they could ride your hand, and if your hand touches any part of your face, that's how it gets to you. Or they can be, if there are any of those droplets that are floating in the air. It's a really, really good question. I love that. And there's different ways that kind of viruses are exchanged. Some of them aren't exchanged on droplets, exchanged on other fluids. There's other kind of ways of exposure, I guess we could say, but they themselves cannot move, right? And we don't think of them as being able to fly or like move through the air. Really good question.
I see them walking on the beach.
Right, we wouldn't see them walking on the beach. We wouldn't see them floating along in the breeze. Just before we get to our experiment, I wanted to talk a little bit more about those envelopes we were talking about. We were saying there are certain viruses, they have to have these capsules to hold in their genetic material, their DNA or RNA, and then some of them have envelopes around them. And these envelopes have special little kind of spikes. You can see over here in our enveloped virus, it has these kind of like green spikes. And we would call them like protein spikes that help them attach to their host cells. Dr. Daniels studies how these spikes affect the body, right? How different viruses get attached, how once those viruses are attached, they cause the body to have responses, meaning they cause our body to do what it does when it gets sick, which is to try to fight off that infection. Our experiment today is going to be about the differences between enveloped and non-enveloped viruses and how we can keep ourselves and our cells healthy. I think let's go ahead and we'll take it back to the two of us, Caroline.
Sounds great.
Perfect. And we'll get started on our experiment here in just a second. But I was wondering if you might tell us what supplies we will need for our experiment today, Caroline.
Of course. So what we have right here, I have them all laid out in front of me, is some dough of your preferred type, to play with and mold, something that's moldable and doughy, basically. And then we have sprinkles. Anything that's small and will stick. If you have sprinkles, that's great. You may or may not be able to use rice if you don't have sprinkles.
That's something that you could use.
You could test it out.
Yeah, I would definitely love to see if it works. I know that these are really good because they don't dissolve immediately in water.
Which is perfect.
We need to use water. So we need something that doesn't dissolve. What else do we need?
We also need some butter. Really great. You want it at room temperature so it'll get on there easily rather than cold and not going to stick at all. And then we need some warm water with soap in it. It can be any kind of soap, but I have dish soap in here and I just heated it up in a tea kettle. Perfect. And then if you want, you can have some gloves or have some paper towel nearby because it's going to be pretty messy.
It is, and I'm excited about it getting messy. I was going to say, I do have hand soap. In terms of the warmth of the water, it doesn't have to be boiling hot. In fact, we want it to be safe for us to interact with. So just about as warm or hot as it gets if you turn on the hot knob on your sink.
Absolutely.
I want to say too that Caroline and I are going to be using butter, but you can also use margarine. That's also works. It comes from a plant rather than from it's not a dairy product, right? It's something that's still made out of lipids, which we'll talk about in a second. And it's something that's still kind of semi-solid at room temperature. Yep. We have a really good question from a friend saying, should both of our containers of water be warm and with soap? Yes, and we'll go through this step by step as well. In fact, I am going to go to my experiment cam here.
Perfect, and I'm going to share the links in the chat that Peregrine mentioned earlier.
Oh, perfect. There we go. So I've got my sprinkles. Oh, and I should say too, I have a spoon, which can be helpful as well. But I think
Smart.
we'll probably be okay with that one if we don't have one. So our first step is going to be, we're going to create our viruses. Now, we want to create one virus that is an enveloped virus and one virus that is a non-enveloped virus. Remember how we said between these two types of virus, they have things in common. They all have that genetic material, that DNA or RNA, and they all have a little capsule. So what we're going to do is we're going to make 2 capsules out of our Play-Doh. So I have just a little chunk here. It doesn't have to be super big, right? Just about like this. We want them to be roughly the same size, but they don't have to be perfect. All you need to do is roll them in your hands. And I guess we don't have any genetic material inside of ours, but we can pretend that it is inside of our capsule.
We can use our imagination.
Exactly. So in a real virus, this capsule would be made out of protein. And that's just kind of a building block that, that many things use, including living things, to make body parts out of. I know that you and I all have protein in our bodies that helps build up our different body parts. So similarly, viruses need this kind of protein capsule to keep their genetic information in. So right now we have 2 balls that look pretty much alike here, right?
Definitely.
What we're going to do is we're going to add something to the outside of one of them. You're going to take your sprinkles and you are going to kind of just shake them out a little bit like that. Because what we're going to do is we're going to roll this ball, our little virus, into some of these sprinkles. And what this is going to represent is these sprinkles are different kinds of things that help the virus get attached to host cells, right? So even though unenveloped viruses do not have the same kinds of protein spikes that envelope ones do, they do still have things that kind of help them stick to their host cells, almost like Velcro. So I'm just sticking those on there, and I'm going to roll it in my hand a little bit. Now it doesn't have to be totally covered, but like a little bit covered in some sprinkles. How's yours looking, Caroline?
Mine is getting nice and sprinkly over time.
Perfect. I've got mine pretty sprinkly as well. This virus is our unenveloped virus. So this virus, as far as we're concerned, is finished. It has its, genetic material inside. It's got that protein case, and then it's got some of those things to help it stick to this to its host cells. For our next virus, it has that protein case, and we're going to add something else to it. So what we're going to do is we are going to take our butter or margarine, and we're going to get a little bit and coat our little virus, pretend virus, in this substance. So I'm going to take my spoon. You can also just use your finger, but not a whole lot, about like that much. And I'm putting it right in my hand, and I'm working on spreading it all over my ball here. So this is a little bit messy, but also a lot of fun. You can see mine didn't cover the whole thing, so I'm going to put just a little bit more on the other side as well. How does this feel in your hand, Caroline?
It feels very greasy.
It feels greasy in my hand too, and very kind of sticky almost.
Definitely sticky. And I'm also going to put a little bit more on because mine did not cover the whole one.
That's okay, and it doesn't have to be perfectly covered, I should say, but we want most of it pretty well covered. The reason that we're using this messy substance, right, this butter, is because butter is the same sort of material that the envelopes of viruses are made out of. So butter and margarine, right, are fats. They are derived from either animal fats if they're butter, or from plant fats if they're margarine. And those fats are also called lipids. So that's the name of them. Maybe I'll type them in the chat after I've wiped my hands off. But that lipid envelope is kind of an extra layer that helps keep the virus's protein capsule safe.
Happy and healthy.
Correct. And what we're going to do is, just like with the other one, we're going to roll it in some of our sprinkles. We just want to make sure that these are nice and covered. These are going to represent our protein spikes.
And they're definitely sticking a little better than our non-enveloped.
Oh yeah, they love to stick to these.
Yes.
Once you get your once you've got your protein spikes attached to your enveloped virus, what you can do is wipe your hands. I know that I have got some paper towels here that I'm going to use, because we will be touching this one again, but if you have a spoon, you can touch it with the spoon, and also we won't be rolling it in anything. It shouldn't get all over our hands. What do you think, friends? Were you able to make your 2 different types of viruses? Caroline, what kind of differences are you noticing when we look at our 2 different types?
Oh, I noticed for sure on mine that one of them is very, very kind of easy to catch on you, the sticky one, and that it's covered in a lot of sprinkles and maybe bigger.
For sure. Oh, I noticed that too. It is bigger. It's got this whole extra layer. So this one is naturally going to be bigger than our other one.
They started out at the same size, but that extra envelope kind of increased.
Exactly. It increased the size. So that's a great observation. And that's the case with many enveloped versus unenveloped viruses as well. What we're going to do is we're going to kind of explore another difference between them by testing them in some warm water with soap. So I'm going to put these aside. They're going to be just right here. And I'm going to prepare my 2 containers. Still got a little bit of sprinkles everywhere, but that's okay. So I've got a little bit of warm water with me. I'm just going to put one in each of my container. And again, you can see that this water is pretty warm, but it's not water that is going to hurt my hand if I touch it. This is just kind of regular warm water. I did put mine in a kettle, but you can just use the water that you get from your hot water tap if you've got that. So we have some warm water, and I'm also going to grab some of my soap, and I'm going to put 2 pumps in each one. Whoops, well, if I can pump any out, let's see. There we go.
Got this.
2 big pumps out. And I'm going to give it a little bit of a stir just to kind of make sure that it's dissolved. Maybe I'll even do one more since my pumps were kind of tiny.
Yes, and I already have my soap in here. You can add yours and stir it in anytime.
I see that it is so nice and foamy. That's so cool. So friends, I think you might have an idea about why we're using warm water and soap. What do you think what do we use warm water and soap for when we're talking about keeping ourselves safe from things like viruses? What do we use it for? What do you think?
Got a great response in the chat here. Washing hands.
Exactly.
Yes, absolutely.
So we use soap and water to wash our hands, and actually it's an interesting thing about using warm water versus cold water. This is something that I learned when I was testing this experiment. We'll talk about it in just a second. I think I'm going to get ready to test it. Ooh, we have a person in the chat suggesting that we use soap and water in order to kill viruses. So let me ask you this. What do you think will happen when we take our different viruses, enveloped and unenveloped? What do you think will happen when we put them inside of our containers? We're going to put one of them in each in just a second. Caroline, do you have any hypotheses about what might happen?
Ooh, I definitely think they will react differently because they have 2 very different outsides. One's very fatty and one's our protein.
And then we have a person in the chat saying that maybe they will dissolve. This is a great hypothesis. Let's find out. What we're going to do is we're going to dunk them in at about the same time into our bowls here. And if yours are like mine, they sunk. I don't know if you have a different kind of, like I know some kinds of modeling clay will float.
Mine
But either
definitely
way,
sunk as well.
what we want to do is kind of stir them around and see what happens. Now, when we are doing this in the sink, if we're washing our hands with soap and water, how long do we do it? Do you know? How long do you usually wash your hands, Caroline?
I usually wash it to the tune of the ABCs, or about 30 seconds.
Yeah, exactly. Yeah, 20 or 30 seconds is exactly what I do as well. So this is because we want to be able to see kind of a similar reaction as we're seeing here. I'll tell you, it takes maybe just slightly more than 20 seconds for us, just because we're using a layer around a layer for our envelope that's several million times thicker than the envelope of a virus.
True.
But if you're doing this along with me, can you tell me what you're noticing?
And you can also screens if you're just following along and tell us what you're noticing with our viruses.
Yeah, for sure. Ooh, we have a great observation in the chat. We have a friend saying that the sprinkles are falling off. And oh, the one with the butter, a lot of the sprinkles are coming off. I would agree with you. I don't know if you guys can see how many of the sprinkles have come off.
Absolutely, and same here. The sprinkles in this one, there's not much to be seen, or I guess if you swirl it around, you kind of see them swirling around in the water.
So what I'm going to do is I'm going to take our viruses out. So here we have our non-enveloped virus. This one did not have any butter on it. And here is our enveloped virus, which used to have butter on it. But what do you think, friends? Do you see any of that butter anymore?
I'm going to go ahead and do the same.
Yeah, I think all of the butter ended up in here along with all of those protein spikes, right? Now, you'll notice that I did have some little pieces falling off of my non-enveloped virus. But for the most part, those proteins that are going to help it stick to a host cell stayed on for this one.
Same here.
For this one, though, I mean, this one's hanging on by, by a thread almost. But they all kind of fell off when we disrupted, when we kind of broke apart that layer of butter or that layer of fat, right? And this is true for unenveloped and enveloped viruses in the real world too. We use soap and warm water because it helps kind of melt away the outside of these viruses. It renders it inactive. This virus would not be able to infect anyone because we've kind of taken all of the protein spikes away, we've taken that envelope away, and this one cannot survive very well, or it can't infect anyone. It doesn't have the ability to do it anymore since we took away that envelope. Now, the thing about this type, right, this is what we'd refer to as, you know how a lot of soaps and also hand sanitizers say kills 99.9% of germs. This is the like 0.1%. But the thing about washing with soap and water especially is even though I can't make this inactive, it helps me to get it off of my hands, right?
Very true.
Yeah, so washing with soap and warm water, even if it doesn't kill all of the potential virus on my hands, it gets it off of my hands, right? Soap is really good at kind of detaching things. That's the same reason why it's good at detaching stuff like dirt from your hands. I'm gonna go ahead and bring my FaceTime camera back here. There we go. So I've got my two viruses here. Both of them didn't do very well against our warm water and soap test, but especially our enveloped virus, right? This one is totally inactive. And
Definitely.
This one, while it's not totally inactive, we would still be able to wash off of our hands, right? It's kind of slippery, right? Just like a real virus, it would slip right off of our hands. One of the things that I learned was how much of a difference the temperature of the water makes, right? I always thought, well, as long as you have soap, you can use cold water. I can tell you, friends, if you test this with cold water, the butter takes a really long time to melt, even if you use plenty of soap. So what this taught me is that for the same reason that butter melts faster with warm water is more effective against enveloped viruses. So we have done a cool experiment today. I would encourage you to replicate it at home, see what kind of different factors affect it, see if using different kinds of soaps might affect it. That's something that I have yet to test, but would be interested in doing. That is all the time that we have for this week, but I've really enjoyed talking about virus and stuff with you guys. I've also enjoyed highlighting the excellence of Dr. Christine Daniels, who you can all learn more about. So I hope you'll join us next week as well, friends, when we will be highlighting more Black excellence of current scientists in the area and doing some experiments with cyanobacteria. That's going to be a really fun one. We'll see you later. I hope we'll see you next week. Thanks so much, friends.
Thanks, everyone.